Welcome to Mr. Chalk's Revision Tips. In this video, we will look at mitosis and the cell cycle. So mitosis is a type of cell division. Mitosis occurs wherever more cells are needed. It produces two new cells that are identical to each other and to the parent. The process of growth and division is called the cell cycle. So in mitosis, two cells called daughter cells are produced, each identical to its parent cell. So when looking at cells with microscope length, or with microscope, the length of the different stages of the cell cycle can be estimated using this formula. So the length of the phase is equal to the number of cells in that state divided by the total number of cells, and we can times that by the length of the time of the cell in that length of the time in the cell cycle. First part that we need to consider when we're thinking about mitosis is DNA replication. So DNA replication is the process by which a cell makes an identical copy of its DNA. This process is performed at the beginning of the cell division so that the cell divides. Each daughter cell will inherit an identical copy of the DNA. So the requirements for DNA replication are that the original DNA template. So DNA is a double helix made of two complementary strands. Each strand can be used as a template to create a new DNA molecule. You need 3 DNA nucleotides, so these are needed to form the new strands. You need DNA polymerase, so an enzyme that adds new nucleotides to a growing strand of DNA. And you need primers. So these are needed to start the process because DNA polymerase can only add nucleotides to an existing strand of DNA. So chromosomes contain DNA. So if we're describing the function of DNA, it's to act as a code that's going to code for a sequence of amino acids which will go and form a particular protein that will be able to form a particular function. So we're looking at the physical process then of mitosis. It can be split into more or less 6 stages. So the cell spends most of its life in interphase. So during this phase, the cell grows to its maximum size and performs its normal functions. The DNA condenses into chromosomes. So each chromosome eventually can be seen to consist of 2 strands of chromatids joined at a central centromere in an X shape. So the nuclear membrane then disappears and spindle threads form from each of the poles. Chromosomes lie then aligned on the equator of the cell. So each chromosome is attached to the spindle fibers at its center. So the chromosomes appear in a straight line across the middle of the cell. The center of the chromosome splits, so each chromosome divides into 2 as the spindle fibers go and contract. And as the spindle fibers contract, they pull a full set of chromosomes to each end. A new nuclear membrane forms around each set of chromosomes, and the cell splits into 2 genetically identical daughter cells. So if we're looking at why the ability of body cells to divide is important, key things to talk about is for growth, repair, producing sex cells, things like that. If we're looking at what happens to each chromosome is doubled or replicated. And if we're looking at what moves the chromosomes to opposite ends of the cell, that would be spindle fibers. The cell cycle, or cell division cycle, is a series of events that take place in a cell leading to its division and duplication of its DNA. To produce 2 daughter cells, it must undergo or move through the cell cycle. So in bacteria, which lack a cell nucleus, the cell cycle is divided just into 3 periods. So if we go and look at the cell cycle and the stages, G1, also called the first gap phase, so the cell grows physically larger, copies its organelles, and makes the molecular building blocks that it needs in later stages. We've got the S phase. So in the S phase, the cell synthesizes a complete copy of the DNA in its nucleus. G2, so this is the second gap phase. So the cell grows more, carries on making more proteins and organelles in preparation for mitosis. The M phase, or the mitotic phase, is where the cell divides its copied DNA in the cytoplasm in order to make two new cells. So it's where mitosis is taking place. And finally, cytokinesis is where the cell is physically splitting into two new cells. So, in which phases does the cell continue to grow bigger? It would be in G1 and G2, or gap 1 and gap 2. If we're looking at the phase in which new DNA is synthesized, that's going to be the S phase. And what is the correct term used when a cell splits into two new cells? That would be cytokinesis.
Thanks for watching.